Good afternoon, and thanks everyone for joining our discussion on cell therapies. I'm Gula Lifshitz, a biotech analyst at Chardan. So the cell therapy space has been showing some momentum recently. Over the past year or so, we've seen expanded approvals of CAR Ts in earlier line cancers, growing activity for cell therapies in autoimmune diseases, a number of new engineered hematopoietic stem cell therapies reaching approval, including the first genome-edited therapy and the first approval of a cell therapy for solid tumors. However, we've also seen some large cap players struggle to keep up with demand. And we've seen issues with out-of-spec products. So for this session, we have participants from companies developing different kinds of cell therapies, patient-specific and off-the-shelf, hematopoietic stem cells, T cells, and K cells. So we'll discuss issues relating to manufacturing in the space across the board, including what's gotten better and what are the challenges. So I'm joined by Dr. Greg Rosati, Chief Technology Officer and Manufacturing Officer at Century Therapeutics, and Robert Ang, President and CEO of Bohr. I thank you both for joining us today. So to start off, I'll invite each of you to briefly introduce yourself and your companies, including the types of cell therapy products that you're making. And we can start with Robert and then go to Greg.
Great. Thank you very much for having us, Gula. Vor Bio is a cell therapy company actually making multiple modalities, but our objective is to cure blood cancers, particularly acute myeloid leukemia, AML. And we're doing this through two different means. We are making CAR T cells, but actually the CAR Ts that we're making, we're actually using healthy donor-derived cells to make these CARs. And so we really focus not just on the CAR construct, but also the cell phenotype, really aiming for cells that potently expand and persist in patients. Secondly, we have a unique stem cell approach where we are shielding stem cell transplants from targeted therapy. And so essentially what we're doing is making a stem cell transplant, which is standard of care in AML, but making it shielded against the targeted therapy by removing particular epitopes, starting with CD33. We're using genome engineering techniques similar to what has been done in the sickle space already, but silencing CD33 and therefore making these cells hopefully totally immune to CD33 targeted strategies such as ADCs, or in our case also our CAR T therapy. And it's great that we're already in the clinic with both programs and looking forward to important data releases later this year.
Hi, Greg Rosati, Chief Technology and Manufacturing Officer of Century Therapeutics. So Century Therapeutics is developing iPSC-derived immune effector cells, both NK and T cells. And we start with the iPSCs, and we do a series of genetic edits on our cells, and then pick single cell clones, which we isolate, and then from there we create master cell banks. We are in the clinic with a NK type product, an NK-based product derived from iPSCs. And we are having two clinical trials active, one in lymphoma and another in lupus. And we believe our cell is the most highly edited cell in the clinic, has 6 edits. So it has a CD19 CAR, has a safety switch, has an IL-15 producing gene to help persistence. And we have 3 what we call alloevasion edits. So we've knocked out HLA-1 and HLA-2. And in this first series of alloevasion approaches, we have HLA-E as a marker to prevent attack from NK cells, and we're looking at further iterations of alloevasion edits for future programs. And my role at Century as Chief Technology and Manufacturing Officer is to run the technical development groups, including process and analytical development, as well as clinical manufacturing, quality groups, supply chain, et cetera. And we do have in-house manufacturing, which I'm sure we'll talk about later.
Great. So from a big picture standpoint, I'd like to understand how each of you would characterize the state of cell therapy manufacturing today, again, in general, and as it applies to the specific modalities in your platforms. Maybe I'll start with Greg.
Sure, happy to. I think it's different in the different spaces of cell therapy. So in autologous, it's beginning to mature, I believe. I was in autologous until 2019. I was at Celgene, now BMS, from 2006 to 2019. And I can't say a lot about how much autologous has evolved since then, but I'm sure people are trying to find ways to automate and simplify, because it is a very burdensome process, labor intensive and costly. So what we did at Century is a different approach, and other companies are doing the same thing. We're trying to come up with allogeneic processes, which will then streamline things more. And I think we're all still learning quite a bit. I think one of the advantages we have with iPSC-derived cell therapies is that we can really pharmaceuticalize them in the sense that, like I mentioned earlier, we start with a master cell bank and we manufacture our batches from that. So we start with much more consistent starting material than a donor-derived allogeneic, and certainly more than an autologous. So that takes a lot of variability out. And then because our cells have unlimited proliferation potential, we can scale quite a bit. And we're trying to learn from our experiences, personal and also indirect experiences in the biologics and vaccine fields, to take the expertise in terms of bioreactors and harvest operations and filling and apply it to cell therapy. So I think we're really well positioned, but yeah, the field is pretty young, and I think everyone is still learning and trying to get better at this.
Yeah, so I agree with everything Greg has said. I think it does depend on the modality and the starting material. Similar to Greg, we start with healthy donor-derived material, which decreases at least some of the variables when we manufacture. I wouldn't say it's necessarily all, because not all healthy donors are made equal. This is, I think, just to set the stage, what we're doing is probably the most complicated kind of therapeutic, in our space right now, and not just because it's difficult to characterize this material from the back end. It's unpredictable actually what happens in vivo, and what we really want to happen is for reliable efficacy alongside very predictable and minimal safety profile. And that's not always possible, particularly with a drug that is designed to expand multiple-fold in vivo, as well as hopefully persist for weeks to months to years. And so, from that perspective, it's tricky. I would say a few things. Firstly, we've come so far just in the last 2 or 3 years, and even the bigger players like the Novartis's, BMS's, Kites of the world, have learned so much about the process from where it was, where it is now, to increase manufacturing success, to actually figure out what's really important to drug product. I think we've become a lot more understanding of what drives efficacy, which is not just plugging a car into a cell, and actually really understanding that the soldier needs to carry a gun, but the soldier himself needs to also be the right phenotype for good things to occur. And then to do this on a reliable basis, all with the background that FDA is still learning about this too, and is still thinking about the different criterion that are important for effective therapies on the market. So I think it is shifting sands, but we've come a long way, and I think even in the next 2 or 3 years, we'll come a long way still.
Right. So I guess just in terms of things that you may have learned from the autologous CAR T space, you alluded to a couple of those now. Anything else that that the field has gotten better at doing in terms of specifics, or like areas that you think we've leveraged knowledge in the autologous space that can be applied to these more advanced or more complex programs? Greg?
Sure. Yeah. Well, I think we're all trying to learn from the clinical experiences there, and therefore try to make cells that are tailored towards the problem. So that's one of the advantages of doing multiple edits, is that if we could understand what the bottlenecks are, whether it be homing or antigen escape, we can do more things like dual CARs, for example, or put on markers that will help get the cells where they need to go. So, but again, I think we're still young in this space, but I think we're all trying to learn and improve and iterate. Certainly we iterate within our own programs on our own products, but I think we're all iterating together on all the learnings out there.
Yeah, I'd agree with that. There's certain things that we're focusing on, not just what we're doing in the clinic today, but also next generation approaches. So one is that I think to some degree we've been spoiled by CD19 and BCMA. that these are relatively easy targets from the perspective that even malignant cells very highly express these antigens and are relatively easy to target for a CAR T. That's not the case with all molecules, right? So I think particularly as we're getting down to 1,000 copies per cell or fewer, thinking about CAR T sensitivity to killing these populations is something that we need to be really thoughtful about. The second is around we've seen FDA already alerted to semi-random insertion from lenti constructs, black box warnings that's on every product on the market right now. I think we need to think about what the next step is beyond this, using directed insertion. I think particularly as people are migrating into the inflammatory space, which Century and other folks are doing, and I think even within oncology, trying to get reliable targeted insertion is a good kind of long-term thing to focus on. I think lastly, if I were to come to any patient and say, hey, you know what, we've got this great therapy, it's experimental, and what I can tell you is that 30% of the cells that I'm going to put into you are exactly what I'm looking for in terms of CAR-positive cells, but the rest are not. You might be laughed out of the room, but that's what we're facing with autologous CAR Ts today, right? If there's ways of doing things off the shelf, just like Greg's doing at Century, or being able to purify cell populations, I think that's also something that is that Vor is looking into, which I think will hopefully create just a higher quality product for patients.
Mm-hmm. Yeah, that makes sense. Greg, it looks like you agree with a lot of those points.
Yeah, no, absolutely. I think everything Robert said makes sense.
Great. So I guess on the flip side, what are some of the current challenges or bottlenecks in your segments of the space?
Maybe I could start there. So I think there is this question about in-house versus outsourced manufacturing, right? And just speaking as a early stage drug developer, we're still in phase 1 with both our clinical programs. We also don't have the resources and scale of a big pharma who can spend hundreds of millions of dollars into large GMP facilities. But we also then see on the flip side, what does it take for, say, a Novartis or a Kite to actually successfully commercialize without without supply constraints, which a lot of them still have. And I think one is just being smart about, you do need to use CDMOs. There's actually much more capacity at CDMOs than there was historically now, than ever before. But also thinking carefully about in-house manufacturing, what you really own and control in the short term. I'll tell you what we've done at Vor is that we've created an in-house facility that's really much more programs that can then generate proof of concept and then hopefully graduate to either a greenfield, brownfield facility that we own and control, or use a large-scale CDMO or potential partner to really help us commercialize. So that's generally the strategy we've adopted, which means we've tried to make the best of both worlds in terms of that CDMO choice. But on top of that, I think another thing that is sometimes overlooked is process and analytical development. So really developing a robust manufacturing process, including release strategies that helps you be efficient from the get-go, such that when it is time to tech transfer this, it doesn't take forever, it doesn't take a huge number of resources, and that you can do it in a way that's very scalable as you look towards larger facilities wherever they are.
Maybe I'll just piggyback on some of that, Robert. I think developing robust processes and assays is critical. And the way to do that is to invest time in understanding your process and understanding your product. So we like to say process characterization starts starts from the day you get ready to go into the clinic for the first trial. It doesn't stop when you go into the clinic, because you want to keep interrogating the effect of the input parameters on your product attributes. And you want to keep trying to understand which product attributes matter when you look at your translational data and your clinical data. And ultimately, that's how you get a robust process, is to know what parameters really affect which important attributes. And you get robust assays by understanding which attributes to measure, and then interrogating your assays almost like they're each a mini process in themselves. So the more you put into this, the more you get out of it. And there's no simple answer of how much investment you need. The more, the better, I think. And that gives you that robustness that you're referring to, Robert. So I just wanted to highlight that. When it comes to manufacturing, I agree. It's good to have flexibility. It's good to have a plan. It probably depends on your situation. Some companies may not have the time or the money to build their own, especially in today's environment. It's tough. It's a lot of money up front. We were fortunate in 2021 to have our IPO and to have plenty of funding to build our own facility, which we did. We're also fortunate to have a second source. We have a great partnership with FCDI, Fujifilm Cellular Dynamics, who produced product for our first trials, 101. And that worked out really well, because our initial deals with FCDI, which included licensing to reprogramming and differentiation, but it also included our commitment to make our first the first product under the first IND with them. So that bought us time to build our own facility, and now we have the optionality of doing things at our own facility as well as FCDI. So it's great to have multiple sources.
So I guess on the topic of the in-house versus partner, or and in-house and partner, you guys both have experience with both internal and kind of partner development. When do you think is the right time for companies to focus on this? And you kind of alluded to this already, but it would be good to get additional insight there. We can start with Greg.
Sure. Yeah. Well, I think if you have the time, the money, and I guess the third piece you need to have is expertise, because if you have plenty of time, plenty of money, and you build it, but you don't know how to run it or how to hire the right talent for it, it's not going to do you any good. And we were fortunate to have all three back when we built ours. So I think I would do it if you had all that, but in today's environment, it's tougher. So then it's just a matter of being opportunistic. When the funding enables you to do it, you should do it. And just like our strategy now, when we think about pivotal trials and commercialization, we've built a facility that's very flexible. It's actually got the compliance in place to do pivotal trials and even a launch, but we didn't overbuild it. So we have a lot of empty space in there that we haven't built out yet, in addition to multiple suites that are built. So if we need larger suites and want to do a commercial launch, once we get to a point where we're confident in our pivotal data, we'd build that space. Just like, I'd say maybe the answer is when you have a lot of confidence in phase 1 data and you're going to move on to a next trial, there's usually time, because it takes time to complete the trial, do follow-up, start your next trial. That might be the time, if you're with a CDMO, to commit to your own. Or depends on the relationship with the CMO. It depends on your finances. It's really no one answer, I don't think.
Yeah, I'd agree with that. And I think similar to Greg, we were fortunate to have raised a good amount of money in 2021, which allowed us to build the in-house facility. What we did, and our intentions were the following. One, we never aspired to be a commercial manufacturer, so it's a modest facility. We've got 4 clean rooms, but I think importantly that these clean rooms are co-located, actually even on the same floor where our process and analytical development occurs. And so literally the tech transfer is, a few feet away. And to have that benefit is a huge luxury for us, such that, we can develop or modify a process, and we can communicate that and implement that, essentially immediately within FDA regs, of course. I think the flexibility is definitely a factor as well. Our first CAR T product that was dosed in our clinical trial actually occurred over the December holiday break. And we had people here literally Christmas Day making a therapy that would make their way into patients. And of course, our people are extremely motivated to do that. Not something you would ever find at a CDMO, right? So I think that's not to be dismissed. And then lastly, again, like owning and controlling this process, particularly in the early stages of product development, is so key. We've already implemented several manufacturing changes, both for our CAR T as well as for our stem cell product, that we've been able to very efficiently tech transfer. And I think, of course, the process will continue to evolve, even potentially into phase 3, but hopefully those changes will be smaller. And owning and really controlling that at the earliest stage, we think is strategically very important. So I think outside of that, Gula, look, if you're forced to use a CDMO, we have been using a CDMO as well for our stem cell program, and developing a trusting relationship there is really key. So someone who you believe is actually looking after your best interests, shows flexibility, shows the willingness to work with you as you're making changes, I think is really key, if you cannot have that luxury of an in-house facility.
Yeah, and I just would add, and the right expertise, right? Because not every CMO has the same set of expertise. So depending on what your product is, your type of process is, it's important to find a good fit or someone who's got enough experience and the ability to learn, and then stay committed to you, as Robert said.
And so, Greg, the relationship with Fujifilm has also been positive in that way for you guys.
Yeah, especially I guess to that point I just made about having someone that knows how to run your type of process. We co-developed the process with them, and we licensed the initial differentiation technology, which we use for our 101 product to make NK cells, from their starting process. Of course, it evolved, but it evolved together. So it really was a co-development effort, which helped a lot too. And I think a lot of CDMOs would be willing to co-develop, but I guess the point is you have to be able to co-develop in a way that's obviously beneficial for you, the IND sponsor, but also something that they can handle. In our case with FCDI, it synergized really well. So I would just recommend being really diligent about what a CDMO can or can't do. And they'll always tell you they can do everything, but find out what they really can do. So we were fortunate with FCDI because they were experts in this for a long time.
Is there anything that either of you would do differently?
Look, I don't think so, not right now. I will say that when Vol was first starting out, we had chosen a CDMO for our cell therapy manufacturing that we're one of the bigger vendors out there, who is well known. We decided to switch and move to a smaller CDMO. I think firstly because we weren't quite getting the responsiveness that we wanted. This is even before we'd even created a process, to be honest. But I think there was some early signals there, and I would also say that during the initial part of the pandemic, we had heard that CDMO had basically shut down for 6 months, versus the CDMO that we eventually chose, which is called C3I. It's actually the GMP manufacturing arm of the Maisonneuve Hospital in Montreal. They showed incredible flexibility for us, with being able to never shut down and actually staff this up during the pandemic, and allowed us to get into the clinic very quickly. And so I think the decision to move to a smaller CDMO, but someone who still has scale, has that expertise that Greg talked about, but really displayed the flexibility for us was really key.
So not all CMOs are equivalent for a particular company's needs.
Very much.
Definitely true.
So on that note, actually, we have heard from a number of CDMOs at this summit that have been talking about their growing expertise. Can you expand a little bit on the capabilities of the CDMOs in this space more broadly, from your experience here?
Yeah, maybe I could start. So I think, again, we make 2 separate cell products, but ultimately we'd love to make both products in the same facility. But manipulating hematopoietic stem cells is very different from manipulating T cells, right? T cells you can culture out, and they are built to replicate. HSCs are built to be quiescent, and tend to be more fragile when you manipulate them. And so really understanding capabilities for both modalities is absolutely important to us. We use specific instrumentation. I think these are relatively common in industry right now, but really good experience, not just using these instruments, but actually being able to tweak these instruments and maintain them over time is really critical. We are also moving into semi-closed systems that help us, scale this even further. And so experience there with some of these different kinds of facilities really important to us as we're thinking beyond the immediate scale and going into commercial.
If I can jump in on this question, if you like. I don't have a lot of experience at CDMOs, FCDI. Like I said, we work with real closely, and I can speak very highly of them, and I'll come back to that in a second. I think I was fortunate at Celgene, which before it became BMS, we pretty much did everything in-house. Our partners, Bluebird and Geno, had some of the vector made outside, but I didn't really work on that side of the business so much. So I can't speak too much about other CDMOs, other than knowing that some have certain expertises, others have other expertise. But when it comes to FCDI, it's just a great example of having the expertise in iPSC reprogramming and iPSC differentiation. These are really, really complicated technologies. I've done cell culture my whole career. Cell culture has its own idiosyncrasies every time. iPSCs are particularly challenging. So I think having a partner like FCDI is so beneficial because their breadth and depth of experience is so great. So their success rate is going to be naturally very high. So really, really pleased with that. We're really lucky to be able to work with them and continue to work with them. So I think when I say to someone, look for a CDMO, those are the kind of things you want to look for.
Got it. So we've talked a little bit about automation and a few innovations that have been alluded to during the course of this conversation. But in general, science moves pretty quickly, but it does take time for new scientific technologies and insights to make their way into manufacturing processes and eventually into clinical programs. So are there any innovations around the corner that you think could address some of the current challenges that we discussed earlier?
Yeah, I mean, I think firstly, I think there's a general philosophical approach that we take that we really highly value process development and analytical development. And so we have a department that we've built out here with really key expertise. And we happen to have actually structured that group to report into research. I think more common in the field is for that group to report into manufacturing. I think there's pros and cons for both structures, but what the benefit of reporting into research allows us to really embed process and analytical development right where the hardcore science is occurring. And they can see that and really help translate that hardcore science into a robust process that can make its way into a GMP environment. And so really kind of grounding that the science handoff actually doesn't occur until the GMP environment. And that approach has really benefited us, over the last few years.
Yeah, maybe I'll just take a different direction if I may. I think when it comes to innovation, we feel really good about one of the key ththings for us with iPSC is we have to scale up. To get the benefit of the platform, we have to scale up. We have to drive down cost of goods because we can. So we need to be able to do that, especially in autoimmune where the demand we expect to be so high. So that'll be a big advantage for us. So when it comes to scaling up bioreactors, we feel really good about that. I think all the technologies out there from the biologics and vaccine worlds, I have a great team of bioengineers and scientists, many of whom worked in those fields previously or worked in cell therapy scale up previously in allo. So we feel confident we can do that. I think the piece that's needed that I know there's particular players outside of our company working on that we want to partner up with, which is filling and visual inspection. We're going to fill thousands of vials and you have to visually inspect them all. So you can do that manually or in a semi-automated fashion, but automating that will be critical. And I think there are technologies out there in terms of imaging where you can replace human eyes with imaging systems to determine whether there are extraneous particulates in your vials. So I think we don't need it tomorrow, but in the next few years, we think we're going to need something like that. And I think the field has it, but I don't think it's maybe being put together really for us just yet, but we're going to be at the forefront of that. And I'm sure other companies will be too.
Yeah, I think it's also worth thinking about what the regulatory authorities and what's acceptable from that front. For us, there is a balance here, right? So if we want to make a process improvement, we also have to think about, well, how would we actually implement this? Does the current IND provide us like flexibility that we can just put in the annual report? Or do we actually submit an amendment? What kind of data do we think we need for that amendment? What kind of questions do we think it would trigger to FDA? I think there's certain things that we'd want to do that basically it would be a new IND. I think that's fine. We have to make a decision whether we want to do that versus, actually file an amendment and really try to push the envelope in terms of what we can implement in the clinic today under the existing INDs that we have.
That's a great point about if you can write your IND in a flexible enough way that things can just fall under annual updates, it's really important. So good advice. Don't lock yourself in and be so prescriptive, but you can't be so general either that the FDA says, well, what exactly are you doing? So it's a real art to get that balance right.
Yeah.
Exactly.
Yeah, it sounds like getting the balance of giving yourself sufficient flexibility in the future to incorporate things that you have line of sight to potentially needing to move things forward. So I guess staying on the regulatory topic, we are seeing cell therapy products become more complex, for example, with multiplex edits or multiple CAR constructs. So what does this kind of complexity add to the CMC equation? Greg, we can start from you.
Sure, I can start there. Yeah, I think when you do all these edits, and in our case, start with an iPSC, there's going to be quite a high bar we have to match when we bring our product forward to IND. And we talked to the agencies about this, and we have a pretty good handle on what to do, and obviously did well with that, getting the IND through. But you definitely need to make sure that your gene goes where you say it's going to go. You need to make sure you don't have off-target integration. And when it comes to all these edits and also the fact that you have iPSCs, you need to think about variants that might arise and what these variants mean. So you need to study a whole genome in a number of ways. The FDA has been very clear about whole genome sequencing being something they want you to do, not necessarily report to them and give them all the data, but do it, understand it, and use orthogonal methods to look at things, because it's not so straightforward that you know exactly what's going on. You need to interrogate the data and understand what might be happening, and then do risk assessments around that and say, okay, I have certain variants, but are they presenting risks that are not tolerable, or are they risks that I can live with? And then you present that to the FDA. And we've done that and brought quite a bit of rigor to the table, and we feel really good about that. But it's something that we continue to learn about, continue to streamline, and try to become a leader at that, because it's something we have to do with all the edits we have.
Yeah, look, I agree with Greg there. And look, I also think that standards are changing. So one example is that we saw the very first CRISPR engineered approved product now, come to market for sickle and beta thalassemia, which is amazing. To think that we have a curative product that is, deliberately genome engineered. The safety package regarding off-target editing for that program was scrutinized. I mean, we actually saw, FDA hearings talking about this. There was a natural variant. There was a concern. But I think they proved to the world and to FDA that deserved an approval. Now, that size of that safety package was actually smaller than the size of the safety package that we submitted in our IND. And that's because standards have shifted, right? And the amount of science that can interrogate some of these questions has also shifted. What they were doing in the clinic, 5 years ago is very different to what, can be happening now. I only say this because that our science and understanding of these things is advancing quite rapidly, and the companies like ours really need to keep up and make sure that safety hurdle is being met. I would also say, my bias here is that FDA right now is actually maybe hyper concerned about things like off-target editing, oncogenicity. I think that there's probably going to become a bit more of a balance soon. We're really understanding the context of treating patients with, malignant disease that, is inevitably fatal, and really understanding what those risk benefits are versus kind of a moratorium saying, hey, any off-target is bad.
Yeah, it's a great point about the fields continuing to advance. I think we all have to be very open-minded about what tools there are and constantly evolve our thinking, learn from each other. And I think, like you said, Robert, you have to assess everything, and nothing is black and white, right? You have to decide what makes sense. Probably depends on the indication you're treating, right? So the risk benefit is always something you have to weigh.
Yeah, absolutely. I think even in the AdCom that we've seen for the CRISPR program, I think a lot of what came up was the kind of risk benefit for a particular indication and things like that. So that is in line with what we've heard from regulators and also just company feedback thus far. So I guess sticking on the analytics theme, or kind of understanding what is in your product, what kind of analytics do you use to understand your product during and at the end of manufacturing? And again, what are the important parameters from your perspective? And I understand it's probably going to be different for the different types of products that you're advancing. So Greg, we can start with you.
Well, there's a whole bunch of things you're looking at. So obviously we have edits, so we need to make sure the edits actually are working, that we're expressing HLA-E, that we're expressing our IL-15 gene, our CAR, et cetera. We're looking at, of course, cell viability and number. We're looking at certain phenotypic markers that say our cells are NK cells or T cells. And then there's potency, which is a fun one because nobody really knows exactly what makes the cells potent. So typically what I would advise people do is in early phases, you want to do multiple functional assays that all might be indicative of potency, and then gather those data. Maybe don't make them all release assays. Maybe the FDA will want one, but study them all, gather the data. And as you learn from clinical trials, then you narrow down to one by phase 3, hopefully one. Maybe you need more if it's warranted. And then I mentioned the safety assays. Of course, you need to show your product sterile and free of mycoplasma and endotoxin. And when you think about the different stages, master cell bank, et cetera, there's going to be certain need for in vitro advantageous agent testing, et cetera. So there's a whole bunch of tests you have to do. And I probably missed, showing you're free of the genetic editing components. There's probably a few others I may have missed, but that gives you a flavor of the many things we have to do.
Yeah, and you know, I think people may be aware that there is the guidance out there for CAR Ts right now. It is somewhat useful, I would say, but FDA provides certain areas that with some specificity and others that they did not. And so I think there's a lot of things to discuss with the agency as you're approaching an IND. I'd also want to comment that aside from the more, typical in process and release assays that Greg mentioned, you have to be really thoughtful about the assays, the exploratory assays that you want to conduct, because I think we're still figuring out exactly what dictates safety and efficacy for these products, right? And the way that we approach it is, let's imagine we've treated our first patient, and certain things happen regarding in vivo expansion. Do we see GVH? Do we see CRS? Other safety hurdles. What kind of change to the tumor burden are we observing, and when does that happen? And therefore, what would we want to know in the drug product or earlier that might be predictive of some of these things? And at least being able to have those assays somewhat set up such that you can analyze and then later correlate to what happens in the clinic. And so those are things that do require a bit of investment up front, as well as forethought around what could be important later on.
To having almost like translational assays incorporated to help guide decision making.
Exactly.
That makes sense. What are the major cost drivers in your processes, and what are you doing to reduce the impact? Greg touched on this a little bit with the scaling, et cetera. Robert, we can start with you.
Yeah, sure. So I think input material is still, something that everyone struggles with, even with off-the-shelf components. I think, for us, we use healthy donor-derived material, which can be very affordable, particularly if it's, say, a related donor. But what also helps with us is that we can technically make 2 products from one, where if we make a stem cell product as well as a CAR T product, we want it actually to come from the same donor. So that definitely helps with COGS. I would say that it's much more common now to have, and maybe a bit more commoditized around plasmid production, around lentivirus or AAV. I think we still need to think about important process steps like electroporation and how those costs factor in. Just cell media is non-trivial. Cytokines non-trivial. I think over time these costs will come down, but we've still got a ways to go before it looks like small molecule gross margins, for example.
Yeah, I think you hit on a lot of the key points. That's where we're trying to leverage scale up, because think about cytokines and media, for example. Not only scale up is going to help us, but the things you talked about, Robert, optimizing your process and having strong process characterization. If you optimize things, you're always going to save money. And then when you think about something I talked about earlier on my slides in my fireside chat, which is having the right process control. So designing, for example, perfusion systems to take out waste, but also add nutrients as needed. The more you understand what process control with the right analysis of the right metabolites, you're not going to waste media. Because I'm sure most of the processes these days, I'm sure all the autologous processes these days, for example, are wasteful because they're not going to use all the components of the media, but something was developed that worked and people didn't want to change things. They had to go fast because it was life-saving. But you might have 10 times, 2 times, 3 times more of something really expensive that you need. So that's one thing, optimizing. But then when you perfuse, you can give things at the right time, the right way. So I think the more we understand the technology and develop things, the more cost effective it will be. And like I said, I don't want to beat the dead horse, but the scale up, right? If we be an iPSC, being a scalable platform, the most scalable platform, theoretically, we can get the cost of goods down to the lowest value. And our job is to figure out how to do that really well.
Mm-hmm. So in the last couple of minutes, I did want to ask where you guys think the field will be 5 years from now, incorporating all of the advances and dealing with the challenges that we're dealing with now. Will we have solved everything?
I don't think so. I wish.
5 years. Yeah, not 5 years.
Look, I think even just we take for granted that you can make a CAR T and deliver it to a patient within weeks and that order is reliable, right? I think that is non-trivial, just even given today's technology. But I think ultimately we are becoming smarter with the manipulations of cells. We're becoming smarter with the edits that we're making and insertions. I think we're also going to become smarter in just the way that we purify these products and really characterize them. I'm hoping that next 5, maybe I would say 10 years, we're looking at a total sea change in the way cell therapy looks. But unfortunately, just given the cycle times and what we develop, it is one step at a time.
Yeah, we won't have solved everything in 5 years, but hopefully we'll make progress. Hopefully, the durability of hematological treatments will get better. Maybe some cases be curative, and hopefully we'll start solving solid tumors, because that's something that we all want to solve, and I think it's a long way to go. And I think we'll make a lot of progress, but definitely not solve everything, which will keep us all in business for a long time, and we'll keep having this conference for many years, I'm sure, too.
Yep, absolutely. So with that, I think we can wrap up. So I'd like to thank both of you again for the great discussion that we had.
Thank you, Gula.
Yeah, thanks for having us. Appreciate it.